will be given by Dr. Tuohy. Kieran Tuohy is lecturer at the Department of Food Science and Nutrition of the University of Reading and leader of the Nutrition and Nutrigenomics group at the Fondazione Edmund Mack at Trento in Italy. His research interests are focused on dietary modulation of the human gut microbiota, examining the impact of whole plant foods, pre- and probiotics. In the topic of personalized nutrition for improving the gut microbiome, Dr. Tuohy will address the questions, feed it, change it, or swap it.
Okay, thank you very much. And first of all, I'd like to thank the organizers for inviting me here today. It's a great honor to speak to this audience. As the chair said, I'm a microbiologist working in a nutrition group in an agricultural research institute, so I've got a very different perspective on diet, microbe, nutrition interactions than some of the previous speakers, but we're sharing a lot of common themes, even though we're coming from very different angles. So today I'd like to talk about the role of the human gut microbiota, especially in metabolic disease or obesity, and its potential for personalized nutrition, and how we can improve health through feeding it, changing it, or swapping it. So just some very general background to introduce the gut microbiota. We all carry around a very complex collection of microorganisms that inhabit our intestines, maybe up to about 1,000 different species. Many of these are unculturable. They won't grow very easily in the laboratory, and so they demand high-resolution molecular microbiology methods to look at their DNA to find out what they're doing. We have closely co-evolved with this complex metabolic mixture of microorganisms all the way through human evolution. And so we're beginning to see that interacts very closely with many human parameters, with our diet, with the effect of drugs, whether drugs work or not, whether they have side effects. They play an important role in educating our immune system in early life. They play a critical role in gut physiology, providing about 50% of the daily energy requirements for the gut wall and helping the intestinal cells bind together to prevent a leaky gut. As I'll mention later on, they play an important role in the enterohepatic circulation of bile acids, and bile acids are coming up more recently as very important cell signaling molecules in the human body, not just involved in fat absorption. Recently, they've been shown to impact on systemic energy metabolism and how we store energy in edibles tissue, and very recently, impacting on brain development and even cognitive function. So the gut microbiota is becoming up as an organ in its own right, playing an important metabolic and immunological role within the human body. And if you like, we've become an ecosystem with our cells and 1,000 microbial species. These interactions are complex and far-reaching, both in terms of diet-microbe interactions and microbe-host interactions. For example, microorganisms break down complex carbohydrates and polyphenols that reach the colon into compounds that we can then absorb and use throughout the body, like short-chain fatty acids, acetate, propionate, and butyrate. They break down polyphenols into small phenolic acids that also have roles throughout the body. As I mentioned, they play an important role in the enterohepatic circulation of bile acids, deconjugating bile acids and producing secondary bile acids that are then reabsorbed at the end of the small intestine and even in the colon. And they play an important regulatory role in immune training at early life, but also throughout life and indeed into old age. So for me, one of the most important aspects of this interrelationship is how it interacts with our diet. And as we've heard from Professor Sanders earlier on, the dietary recommendations around the world agree that this profile of diets, you could put up the American Eat Well plate or the UK plate, it's a very similar story. Increase intake of whole plant foods, of fruit and vegetables, whole grain cereals, complex carbohydrates, low-fat dairy products, and reduce intake of red meat, highly processed foods, foods with a high energy density, with refined carbohydrates and sugars. Professor Saunders also mentioned the environmental impact of this, and this is very neatly displayed in Italy with the double pyramid brought out by INRAN and the FAO, which shows the environmental impact of this dietary profile, the modern Western style profile, compared to the more traditional dietary profile that we're recommending people take on board. You could also look at this in terms of gut microbiota, because these two different diets will have two very different impacts on the gut microbiota. And that's something I'm very interested in, particularly how the dietary fibers, polyphenols on one side impact on microbial metabolic output, while proteins on the other can have some other activities that might not be as beneficial. Looking at some of the health associations of these diets, this is a study from Molise, the Molisani group in southern Italy, led by Professor Di Gaetano. He found that elderly people are some of the few people in Italy who still have high adherence to the Mediterranean diet. And he found that people who have a high adherence to this diet have a greater health-related quality of life. And surprisingly, the health-related quality of life is greater for mental health and physical health. And looking at the statistics, both and the food composition, both the antioxidant content and the dietary fiber, so the polyphenols and the fibers in the food independently explain these relationships. Of course, it's not related to geography. This kind of diet is a traditional diet that's been used in many parts of the world. Professor Sanders also mentioned the Japanese diet. In Okinawa, a small island off the south coast of Japan, they have the longest lived human population on Earth, living up to about 83.8 years on average, compared to mainland Japan, about 82.3, and the US with 78.9 years. If you look at chronic disease risk, this population in black have lower incidence of cancers, of cardiovascular disease, colon cancer, prostate cancer, breast cancer, and lymphoma compared to mainland Japan or the US. And they put this down to a closer adherence to their traditional Japanese diet, which is high in fruit and vegetables, high in soya, high in fish, and high in fiber, but importantly also low calorie intake throughout life. So the energy balance, the energy density of foods is critically important. In Europe, we have the EPIC studies, which followed about 75,000 Europeans in different countries and found that adherence to Mediterranean-style diet in elders was associated with lower all-cause mortality. In Bile in the UK, we found that elders who ate more fruit and vegetables also ate more fruit and vegetables in early life, in childhood. So if you want to intervene, you have to start early, as we heard from Professor Fecal. For me, two of the most important parts of joining these different diets are dietary fibers and polyphenols, because for me they impact on gut microbiota. Dietary fiber provides the energy and the carbon source for the majority of gut bacteria, and 90% of dietary polyphenols are not absorbed in the stomach and small intestine, reach the colon, and there are transformed by the gut microbiota into smaller compounds that can be absorbed and impact on human health. So looking at this more closely, you can imagine the gut microbiota being supported by fermentable carbohydrates on traditional diets where you have high fiber intake, producing short-chain fatty acids, and we know that these small organic acids impact on things like energy homeostasis in adipose tissue differentiation and epigenetics, gene expression, gut hormones, in satiety regulation. On the other hand, a Western-style diet deprived of dietary fiber, the gut microbiota switched to amino acid fermentation to get their energy. And the end products of this type of fermentation is very different. You get amines, indoles, ammonia, sulfides, and nitrosa compounds, which are variably genotoxic, carcinogenic, cytotoxic, and some of them, like trimethylamine N-oxide, have been directly linked with arteriosclerosis in humans. So again, looking at obesity as a case study, when we shift from traditional diets to more Western-style diets, and this is happening around the world, we see an increase in obesity as this dietary change is happening. In Italy, a Mediterranean country, traditionally has been associated with low rates of obesity, but you can see childhood obesity in Italy now has the third highest rates in the world after Greece, the United States. Italy is in third place. So in the Mediterranean countries, childhood obesity is becoming a particular problem, again, as we heard earlier. Over the past 10 years, a new player in terms of gut microbiota has entered the stage, and this is the gut microbiota. This was seminal work by the University of Washington, Gordon's group, that showed that obese people not only have a different collection of bacteria in their gut, but when you put these obese people on a weight-lowering diet, their microbiota changes towards a lean-type profile. Also, if you take that gut microbiota from an obese animal and put it into a germ-free lean animal, that animal becomes obese. More recent studies, as I mentioned, showed that certain metabolites produced by the gut microbiota, like trimethylamine N oxide, when it's produced from choline, or carnitine, which are compounds in meat and fat, can lead to an increased rate of cardiovascular disease linked to a reduction in bile acid pools. So feeding it seems to be an important part of how we can control our gut microbiota. And it makes sense since we've, as I mentioned, evolved with our gut microbiota right down through human evolution. If you consider what we've been eating all the way through this time, it's only up until maybe the last 50 years that we've seen a huge drift away from diets that contained high amounts of fruit and vegetables, whole grain cereals, and were low in refined carbohydrates and sugars, for example. With the advent of the green revolution in the 1960s, access to high-density, plentiful food supply has changed our eating habits, and I think this has had a dramatic impact on our gut microbiota, probably greater than all the other historical changes in human nutrition down through the ages. If you looked at just a dietary fiber intake in different assessments or current populations eating different diets, the Paleolithic, rural Chinese people today and in rural Africa, their dietary fiber is 5 to 7 times higher than the current intake in the US and the UK. You can say something very similar for polyphenols. We're eating less fruit and vegetables now than we used to, so we're taking in less polyphenols. But if you also look at cultivated varieties of fruit and vegetables compared to wild varieties, this is strawberry, wild variety compared to strawberry cultivated variety, you can see that they have about 50% less polyphenols. So not only are we eating less fruit and vegetables, but the fruit and vegetables we're eating have less polyphenols, the compounds we think of as impacting on gut microbiota. The effect of this on gut microbiota in relation to obesity and regulating weight in children was very neatly shown by the group of Liping Zhao in Shanghai with a recent paper where they looked at a radical dietary intervention in 2 populations of children. One, the Prader-Willi syndrome children who are genetically predisposed to obesity, and the others were normal diet-associated obese children. They intervened with a diet that reduced calorie intake by about a fifth, so not a huge radical change in energy intake, but they radically increased the amount of dietary fiber. This is just the energy intake you can see over the 3 months dietary intervention, reduced by about a third. This was mainly brought about by reducing fat intake, increasing carbohydrate percentage, but increasing fiber by about 7-fold and soluble fiber by a 10-fold increase. So a radical change in dietary fiber intake in these children. In terms of metabolic parameters, they saw a significant reduction in body weight over the 3 months in the genetic form of obesity, as well as all these metabolic parameters related to obesity and metabolic disease. The same with the children on the more common diet-associated obesity. So they were able to regulate body weight very clearly with this dietary intervention. In terms of gut microbiota, obviously they also had a very big impact on microbiota composition and activity, with a radical increase in relative abundance of bacteria clustered around the bifidobacteria. And at the same time, a reduction in activities associated with production of this toxic compound, trimethylamine and N-oxide, a tryptophan metabolism and choline metabolism. When they took the microbiota from children who had received this therapy and put it into animals, they found that the animals, after 4 weeks, had a much lower adipocyte size and body fat composition compared to animals who received the microbiota from the obese children before the dietary intervention, showing that when you separate the microbiota changes from the dietary intake and put it into a new system, you can bring about the same regulation of body weight and fat accretion in the animals. So another approach to feeding it could be changing your gut microbiota, and this is something that's been tried for many years now, and of course EFSA have a long history dealing with these cases. And it's fair to say that the history surrounding probiotics hasn't received as much support as received a lot of attention, but none of the cases have passed regulatory standards so far. And for me, one of the critical points of this is that very few probiotics to date have been chosen for specific biological activities. They've been chosen for technological reasons, for reasons of history for use in a company, for example. And very few of them have been chosen for a particular physiological ability or traits. This is one example, one of 2 dietary interventions in people with high cholesterol levels from Canada, where they selected a strain of Lactobacillus reuteri for its ability to hydrolyze bile salts. Their theory was that this would impact on cholesterol metabolism. They'd shown it previously in animals. They then conducted 2 large, medium-sized, randomized controlled double-blind studies in people who were hypercholesteremic and found a 10% reduction in total cholesterol, LDL cholesterol, and improvement in the ratio of LDL and HDL cholesterol. They further found that people who were consuming the probiotic had lower uptake of other fats from their diet, especially plant sterols, showing that the microorganism, probably through bile acid metabolism, was impacting on fat absorption from the gut, which was a new theory and one not linked to cholesterol biosynthesis in the liver, which was a previous mechanism proposed. So as I mentioned, bile acids are becoming up as important cell signaling molecules through things like the nuclear receptors, FXR, the vitamin D receptor and PXR, but also TGR5, which is an important regulator on immune cells. This signaling power of bile acids is greatly modulated by the bacteria in the gut. The profile of these bile acids returning from the intestine, whether they're secondary or primary bile acids, whether conjugated or deconjugated, can impact on the activity of these receptors and mediate different cell signaling potentials around the body, most particularly in bile acid metabolism itself, fat absorption from the gut, and liver homeostasis, glucose homeostasis in the liver. Other studies by Nathalie Delzine's group, Patrice Canny, and Everard in the Catholic University of Louvain have long looked at how dietary fibers can, in a high-fat diet environment, reduce metabolic disease and especially inflammation associated with disease. They more recently have found that when a particular organism, Akkermansia mucinophila, is actually much more underrepresented in obese animals compared to normal weight animals. This organism is also much reduced when you put these animals on a high-fat diet and is closely inversely related to the inflammation you see in obesity. They then took this organism and fed it to animals on high-fat diets and were able to reproduce the effect of the dietary fiber in terms of body weight and lipopolysaccharide leaking from the intestine. So they were able to reverse some of the metabolic parameters and the inflammation associated with a high-fat diet by giving a specific organism that they had found was underrepresented in obese animals. So finally, a newer approach is swapping it. How can we take some of this information and radically alter our gut microbiota? And people have been using this for some time now, fecal microbiome transplant, especially for the treatment of Clostridium difficile or difficile infection. It's an antibiotic-associated diarrhea that persists, that becomes resistant to antibiotic therapy and is responsible for about 40,000 deaths in the UK of elderly hospitalized people each year. So it's an important problem. In the case of fecal microbiome transplant, it's looking at about 80% success rate in treatment of this disease. So it's becoming a mainline therapy for Clostridium difficile associated diarrhea. At the moment, the donors are selected merely on safety, so the absence of pathogens or other diseases. There is no attempt so far to select a good fecal donor in terms of metabolic health or obesity, for example. But I suspect this is coming because this is one study from Willem de Vos's group where they've looked at fecal microbiome transplant from a lean individual to a person with metabolic syndrome and found some improvement in insulin regulation. However, what's really holding back the field is making the link between microbiota profiles, their metabolic output, and the response in humans, the physiological output. And this, together with collaborators around Europe, is what we'd like to do in Fundazione Mac, and probably what most other microbial ecologist groups in Vagan and Cork, for example, are also doing, is trying to link up microbiota through metagenomics and metabolomics with host response in terms of transcriptomics from different types of dietary interventions, which we hope will allow us to rationally select donors for fecal microbiome transplant. So select fecal samples that have particular profiles of short-chain fatty acids or bile acids or bacteria we think of as beneficial, to provide the mechanistic basis for selection of efficacious probiotics that might pass the regulatory requirements in terms of cause and effect relationships when they're tested in double-blind randomized control studies, and provide some of the mechanistic data underpinning why the diets we think of as beneficial, the Mediterranean diet being a classic example of why these diets work in humans. So just to finalize, I'd like to identify the gut microbiota, all of us and our gut microbiota, as an important part of personalized nutrition. And if we can deal with getting the food right for our microorganisms, we might also make some advance in improving our own nutrition. So finally, I'd just like to thank the organizers again, my team in Fondazione Mac and previous collaborators in the University of Reading. Thank you.
Thank you, Dr. Tuohy, and thanks to all the speakers of this section for fully respecting the time. We have time for one or two questions from the audience, if there are any. I'm sure your presentation stimulated several questions for longer discussions. I even have my own questions to address to you. So with this, we can have a break until 15 past, when we will resume the session. Thank you very much.